Hello, and welcome again to another episode of Interesting Stories in the History of Diagnostics. This week, our episode is called Can't Live With Them, Can't Live Without Them. Specifically, I'm talking about bacteria. So this is going to be a two-part episode. We are focusing on bacteria, but different parts will have different focuses. In the first, we're going to talk about the history of bacteria. We're going to go all the way from the beginning of the Earth all the way to germ theory at the end of the 19th century and the identification of specific bacterial pathogens associated with specific diseases. And then in the second episode, we're going to talk about the ways in which we were able to diagnose those diseases. How did we detect them? How did we identify them? And then where that's all gone over the course of the last century or so, and where we are now, where things have gone. What are the trends in our ability to detect and diagnose bacterial diseases? The Earth formed 4.5 billion years ago. From about 4.5 billion to about 3.8 billion, we were going through what was called the late heavy bombardment phase. At that point, the Earth was still forming. The Earth and the Moon had split. There was a lot of material from space crashing into the Earth. The oceans formed about 4.4 billion years ago. And then we started finding evidence in the last many years of microfossils as early as 4.3 billion years ago. So just a short time after the oceans formed, about 100 million years, here is evidence of biological life, remarkably early. Some of the earliest direct evidence for the presence of bacteria are structures called stromatolites, particularly found in Western Australia. They are basically structures that were created by bacteria. It took a long time for people to believe that's what they really were, but the general consensus today is that that's what they represent. Basically, bacteria have been here with us ever since. It's probably the case that bacteria were the first land organisms of any kind. Since then, bacteria have inhabited virtually every major place that's possible on and in the planet. They are in the oceans. They are in the air. In fact, there can be remarkably large numbers of bacteria. Today, there are about 100 million bacteria per cubic meter of air around the Earth. Unbelievable. They have been in thermal vents. That is probably where they initially developed, eating things like hydrogen sulfide and methane, not photosynthesizing. It wasn't from light that they got their energy. It was from these other materials. Still to this day, they do in many forms. They're also found in the lowest parts of the Earth. The Mariana Trench actually harbors bacteria which can eat oil. And there are bacteria that have been found in core samples taken from the Earth down as far as almost 1,400 meters. Astonishing. Been found in Arctic ice. I mean, all over. Amazing. And of course, they've been found on, in, and around animals and plants. Bacteria are found on and in many animals, most animals, certainly humans. Most of the organisms that we're accustomed to are so-called commensal. We know that the bacteria are mostly found in our gut. In fact, several pounds worth of bacteria in our guts. It was reported in 2014 that there were probably 10 times more bacterial cells than there are human cells, but no longer does anybody believe that. That's been shown not to be true. In fact, it's about the same number of human cells and bacterial cells. There was a paper that came out in 2015 that set the record straight, and it was a paper by Sender et al. And interesting quote within it said that the numbers are similar enough that each defecation may flip the ratio to favor the human cells over the bacteria. It's about 30 trillion human cells and about 39 trillion bacterial cells before a defecation. Now we believe that they're quite comparable in number. People have taken a look at the occurrence of variants of bacteria on our outer body, and I heard an individual named Eric Schacht, who was at Merck and now is at the company Semaphore, talk about a study that his team had done. And they were looking at the occurrence of different types of bacteria on the human body. And he asked all of us to guess where the most diverse spot was for bacteria on our body. And you might imagine where people would normally think, but they wouldn't have thought where that area was. It happens to be the inside of the elbow. Nobody knows why, but there are more types of bacteria there than anywhere else on the human body. Bacteria have been shown to participate in our digestion, of course, everyone knows that, but even our immune system, perhaps even our mental health. Lots of very interesting studies going on in the so-called microbiome, trying to understand the overall relationship between humans and bacteria. The second way in which we live with bacteria is not so obvious. Even 50 years ago, people didn't believe it. But as early as the late 1800s, people were proposing the possibility that some of the organelles in cells, and so-called eukaryotic cells, some of those organelles may actually have been previously free-living organisms. In fact, bacteria. A woman named Lynn Margulis had been pushing this for quite some time, and a lot of people thought she was nuts. But in fact, she is right. Mitochondria occur in both animal cells and plant cells. They derive from bacteria. And chloroplasts in plant cells also derive from bacteria. Kind of amazing. They are the result of what Lynn called symbiogenesis, where a large prokaryotic cell engulfs a small prokaryotic cell, bacterium, and becomes an integral part of what becomes eukaryotic cells with organelles. So we've got all kinds of reasons to be interested in and be thankful for many bacteria. We also recognize that many of the organisms that live on us are potential pathogens. And there are so many organisms which are known to be associated with infections in humans. Some of the many diseases caused by bacteria in humans include, for instance, tuberculosis, cholera, bacterial pneumonia, anthrax, tetanus, gonorrhea, plague, leprosy, and Legionnaire's disease. People have known about these bacterial diseases for a very long time, but they didn't know they were bacterial. But as early as 1025, the physician Ibn Sina actually proposed something like germ theory. And a number of others have over the years, but it really wasn't accepted by anyone until the 1800s, when Louis Pasteur pioneered germ theory in the 1850s, leading to both vaccines and ways of sterilizing food. But then in the late 1800s, Robert Koch and his colleagues looked at this, and they identified the causative agents, the pathogens, the bacteria, for anthrax, TB, tuberculosis, and cholera. So that was an enormous step forward. Before that, the detection of these bacterial diseases was from observations by very astute physicians who understood the symptoms and therefore could identify the disease, but they didn't know what the cause was. They were usually pretty good at it, but there were some exceptions to that. For instance, medical historians have taken a look at some of the symptoms associated with leprosy and came to realize that in the Middle Ages, a number of people with acute acne were probably identified as lepers and put into leper colonies. So we've always needed better diagnostic methods than simply observation. After the identification of some of these organisms as pathogens with Robert Koch and the continuation of that work, people were now very interested in understanding how to isolate and observe the organisms themselves. And one of the major advances there was the Petri dish, invented by Julius Petri. He was actually a member of the Robert Koch laboratory, and he came up with this way to use these glass plates at the time to put in a material called agar and nutrients, and put one plate over the other and create a perfect environment for the growth of bacteria. It's worked extraordinarily well. Everyone uses it to this day. We have more plastic versions of Petri dishes than we do of glass ones now. Some people still like glass ones. So that was able to identify a number of different organisms on different kinds of nutrient materials within the agar. And a great medical microbiologist can take a look at a plate and tell you more things about those organisms in that disease state than you can ever imagine. But as time went on, into the 1970s in particular, we started developing new tools beyond simply culture to identify bacteria. For instance, the development of immunoassays, antibody-based testing systems. I think as time went on, those were better suited to the detection of antibodies themselves than they were for most organisms, with a few exceptions. But in the late 70s, things changed dramatically. So that's the end of part one of our two-part episode on bacteria. The second part, which will be coming up soon, will go through the history of detection and diagnosis of bacterial diseases, what technologies were used over the last century and a half, and where things are going today. Thank you very much. Hope you enjoyed this interesting story in the history of diagnostics. This episode was Can't Live With Them, Can't Live Without Them. And don't forget to tune in for part two coming up soon. This is Mickey Urdea signing off for Halteris Associates. Join me next time